All right, so we started recording. So we're moving into chapter 17. It was last night that I put up two videos on chapter 16. It's a little bit long together. They're about 1 hour. This is to make up for missing class on Wednesday, and I apologize for that. Took about 10 hours for them to fix. It was funny. It was only 20 units without power. I just happened to be one of them for 10 hours. So I do apologize for that. But given our time that we have today, what I want to do is get as far as we can through chapter 17. Today, it's going to be a little bit lecture, but I did find 2 or 3 YouTube videos that I think are more powerful than words. Sometimes seeing is better than like hearing. So part of this lecture is also going to be watching YouTube videos. So usually I try to avoid that, but I think it's actually a useful tool for this chapter. So why don't we move forward? So we're talking about nucleic acids and specifically how nucleic acids, so DNA and RNA, are translated and transcribed into proteins. So things that make up nucleic acids, effectively, there's 3 components. You're going to have a nitrogenous base. Nitrogenous meaning there's nitrogen involved. It's just, don't even worry about that point. It's a base. So probably what you learned in high school about A, T, G, and C, and then with RNA you have uracil, right? Those are the bases. I mentioned this earlier when we were talking about, I think it was chapter 15, right? DNA is made out of sugar. The sugar is the backbone. And that's why we need to expand our understanding of what sugars are, right? So sugars are structural elements. They are these cyclic structures. And so you either have deoxyribose or ribose sugar. And we'll talk a little bit what distinguishes the two, but it does distinguish between DNA versus RNA. That's why you have D for deoxy and R for ribose. And then the last part, what is going to be part of a DNA or an RNA molecule, is it typically has some sort of phosphate attached to it. So a phosphate looks like phosphoric acid. That's where it gets its name from. And we'll see how this plays relevance, but this is where the sequences of nucleic acids actually attach themselves, are all connected through these phosphodiester bonds. And we'll talk more about what that is in a second. So again, you probably learned this in high school. There are sets of bases, right? Bases are going to hydrogen bond to each other, and this is how they match up, and we'll talk a little bit more about that later. But there are 2 classes of bases, and it's based off of their structure. The first is pyrimidines, and pyrimidines you can see as we go through all the structures here, right? All of them look very similar, right? You have this essentially cyclic 6-membered ring where there's 2 nitrogens, and you have that carbonyl oxygen, right? So these are going to be your cytosine, which is C, thymine, which is T, and uracil, which is only used in RNA as a substitute for thymine. And then you have your purines. Purines, again, we can see there's a similar structure, and they're very different than pyrimidines, right? But we see we have a general structure, and these incorporate both adenine and guanine. And so these are going to be our 5 bases. 4 are used by DNA, which are these 4. And then if we look at RNA, RNA is going to exclude thymine and include uracil. And it's one way that your body helps distinguish between DNA versus RNA. We'll talk about double helices versus single helices. But your body can tell the difference between literally one methyl group, which is used in thymine, which is only found in DNA, versus not having a methyl group in uracil, and it's RNA only. So what makes up these nucleic acids, right? I've mentioned this before, but RNA, right? The R stands for ribose, Na stands for nucleic acid. So we have ribose, ribose nucleic acid, right? Ribose is a sugar, and as we see down here, I know I just highlighted yellow with yellow, but hopefully you followed what I was saying. right? But you have a ribose sugar. That is your main base, your main sugar, and then you attach either a base, and then on this side, we're not showing it in the picture, but you would attach a phosphate group. DNA stands for deoxyribose, right? So deoxyribose nucleic acid. And it's not on this slide, but effectively what you lose is this hydroxyl group right here on both. Again, your body can sense that difference of one functional group. It makes a huge difference between functioning as DNA, long-term storage of information, and RNA, which is usually active use of nucleic acid information. And again, I'll explain what I mean by information in a couple of slides. So again, we're talking about the sugar that makes up DNA. This is going to get into a little bit of definitions, right? We're going to talk about nucleosides and nucleotides, and you're literally talking about one letter difference, and we're going to talk about one component difference. So if it confuses you, I really don't think that's a problem. I've even met scientists to confuse the two all the time, but it's good to learn the difference, right? So a nucleoside is going to just be only sugar and base. So no phosphate. So a nucleoside is just a sugar, so a ribose or deoxyribose, and then one of those 5 base pairs we were talking about. I'm not so worried about knowing about an N-glycosidic bond, but we can talk about that. The way the names change, though, is that if you have a base pair, so you've attached a sugar to a base, purines become ocene, so you have cytosine, or you have cytosine, or I'm forgetting the other one, thylacine, right? And then you're also going to have adenine and guanidine, right? So those would be your purines and your pyrimidines. Again, I'm not too worried if this doesn't make sense, but it's good to learn the two, right? Sometimes seeing is better than talking, and so that's what I have here. So again, right, our definition of a nucleoside is going to be a sugar and a base. So my sugar and my nitrogenous base, they're going to come together. So separate, we have adenine and ribose, and then together, the whole complex, that whole thing is called adenosine, right? So when we're going to go one step more, where we're going to attach a phosphate to this hydroxyl attached to the hydroxyl group. And then we'll see the difference between a nucleoside and a nucleotide. Trying to clear there we go. All right. So nucleotides, the only difference, honestly, right? I know there's a lot going on this slide. We were looking at adenine before, adenosine before. The only difference now is we still have our sugar, so we have our ribose seen in yellow. We have our base seen in pink. The only difference is we've attached a phosphate to that final hydroxyl group, right? The way we name this, right? We keep the nucleoside name. So we have adenosine, but we're indicating that, and this is the way you might hear people or scientists or anyone talk about DNA or RNA. You might hear 5' or 3' instead of 5 or 3. The reason for that, regular numbers are given to the bases, and then basically what we said by prime is it's a little bit different. So we're going to have 1', 2', 3', 4', and then we have, sorry, I wrote 3 twice. 4', and then we have our 5' carbon, right? Again, we're counting as normal, like we would do for organic compounds. We started with normal numbers as 1, 2, 3, 4, 5 on the base, because we're talking about the second part of the molecule. We'll call it 1', 2', 3', 4', 5', and that's why we have adenosine 5' monophosphate. Sometimes you might, so this AMP, there's ADP, adenosine 5' diphosphate, and then probably what most people know is triphosphate, so the adenosine 5' triphosphate. to distinguish between RNA and DNA, right? Because we said the only difference between RNA and DNA, and that's actually what's showing it in pink, right? In DNA, let me change color for a second. That 2' hydroxyl is gone. That is the only difference between RNA and DNA. And that's why you see in pink, instead of having adenosine 5' monophosphate, we have deoxyadenosine 5' monophosphate, or DAMP instead of AMP. And then I'll leave it up to you if you want to go through later, but you can go through each of those structures and see how the name changes depending upon the base and then depending upon what's attached to the 5' position. So I know most people think of energy as ATP, but GTP is also just as common. Your body can use, it's pretty effective at using what it can for energy. So ATP is the most common way to use energy in your body. GTP is not as common, but very common for using for energy. But usually, we save that for different courses. All this is a table summarizing how DNA and RNA, so our nucleic acids, are built up. We have bases. Bases are attached to sugars, so they become nucleosides. Nucleosides, like I said, base plus deoxyribose, or base plus a ribose. Nucleotide has an additional phosphate. And then nucleic acid, which we're going to get into, right? It's almost like when we're talking about proteins versus peptides, right? Once things become really long, right? So polymers, so multi-pieces. So you're talking DNA can be billions of letters long, right? So it's very, very long molecule. It sounds absolutely ridiculous to say. It's been proven to be true. Each and every single one of your cell, and you have 1 trillion cells in your body, each of them have 2 meters of DNA. It's 1.8 meters, 6 feet of DNA are packed into each and every one of your cells. That's how much DNA is in each cell. And it's actually quite small. And nature evolved to pack it very tightly. But yeah, sounds ridiculous to say. There's 6 feet of DNA in each of your cell if you laid it out end to end. So we actually went over this naming, so I'm not going to not waste our time, but I'll leave this slide up, but I kind of went through the naming already. So we're going to talk about structure of DNA and of RNA as well. So even though we just kind of think of it as just like floating around in the cell, your body has a very structured way of storing nucleic acid, right? And actually, nucleic acid is incredibly stable. It's not like Jurassic Park when you can literally extract DNA from like amber, but you can extract DNA from things that have been dead for thousands of years. DNA is a very stable molecule. You might not be able to get all of the information back from it, but most of it might stay intact. All right, so let's talk about how nucleic acids become polymers. Actually, maybe I should take a pause for a second. I've said a lot. Is there anything that I could clarify for students right now that I've said? All right, I will take silence as a good thing. Again, this chapter is pretty straightforward. Like I said, we're going to watch a little bit of YouTube videos at the end. I want to make sure we save I think it takes about maybe 5 to 7 minutes to watch the whole thing. So I'll try to make sure we save enough time for that. So how do nucleic acids form polymers? Well, what they do, and I alluded to this earlier, is that you have this phosphate on that 5' carbon of the sugar. What that does, the 5' phosphate makes a covalent bond with a 3' hydroxyl. So it doesn't matter if it's nucleic acid or deoxynucleic acid, because DNA would just have a hydroxyl here, and the same process would happen, regardless if that hydroxyl is there or not. And so that's why DNA and RNA have very similar, at least primary structure. We're going to get into secondary structure and tertiary structure in a little bit, and they do differ. But in terms of how they're connected, DNA and RNA are connected by the same bonds. We define that bond as a phosphodiester bond. So it sounds like a mouthful, but break it down. Phospho means it's through a phosphate. And then I have di, which means two, and ester. So a regular ester looks like something like that, right? So you have your carbonyl oxygen directly, carbonyl carbon directly bonded to the double bonded oxygen, but also through an oxygen. So in a phosphodiester or phosphoester, well, if I just in my mind replace that phosphorus with the carbon, well, that kind of looks like an ester bond. And then this also looks like an ester bond. So therefore, we call it a diester. So the phosphate is now making an ester bond in two places, phosphodiester. And then we typically call it where it's linking up to, and we do it in order of which their connection happens. So again, we're going to do it numerically. So it becomes a 3', 5', 3' off the top sugar, 5' off the bottom. So 3', 5' phosphodiester bond. So take home message, we're connected through phosphodiester bonds. One of them goes through the 3' sugar, which is on the top, and then the 5', which is on the bottom. And just like when we were talking about proteins, right? We said it goes from an N to a C terminus from the same quote unquote direction from like beginning to end is typically called 3' to 5'. In reverse, we call it 5' to 3', like going from a carboxy to an amine terminus on a protein. So we do have, your body recognizes directionality. But as scientists, we also have a directionality in terms of naming things, right? We're talking about from beginning to end, 3' to 5'. And so I've kind of already talked about this, but the only difference here is that this figure is getting bigger and bigger, and we can see the phosphodiester bonds between those 3' and 5' hydroxyl groups. So let's talk a little bit about the structure of DNA. So most people know that DNA is a double helix. The crazy thing is most people probably don't realize is that it was only discovered roughly around when my dad was born. So we're talking about 1.5 generations, or maybe 2 generations at this point, of people who understand that DNA is a double helix. So this was done in the 1950s. We're not talking about a long time. So what leads to this structure of the double helix? So we know there's 2 DNA molecules, or 2 giant strands that wrap around each other. What led to or helped lead to the discovery is that we were able to do experiments, or scientists were able to do experiments and found out that certain base pairs only come in 1-to-1 molar ratios. So that means if I got 50 adenosine, I also have 50 thymidine, or in this case, thymine, because we're not looking at the phosphates. You'd also, if I had 100 guanine, I'd have exactly 100 cytosine. So we had this 1-to-1 ratio between certain base pairs. And this has helped us figure out that DNA it runs anti-parallel, which means one side goes 3' to 5', the other side goes 5' to 3', and they're exactly opposite. And if they're exactly opposite, well, then they match up so that every adenosine matches up with a thymine. And so you probably learned that in high school biology, A to T. And then guanines only link up with cytosine, and that's going to be G to C. They do these linkages through hydrogen bonds. And I think in high school, because I remember learning this, maybe things have got better since I was in high school, they used to tell me that hydrogen bonds is what gives the structure to DNA. That is absolutely not true. If that was the case, your body, DNA would just break down. We don't get into it because we've never talked about this intermolecular force, but effectively, when those molecules start spiraling on top of each other, those bases, and we, again, in this class, we didn't get deep into it, but if you did orgo, all of these double bonds they're projecting into 3-dimensional space additional electron cloud. They're called pi orbitals, and they actually overlap with each other, and they form attractive forces between each other. We won't get into it. H bonds definitely lead to complementarity. That's how DNA can basically make a copy of itself and makes a double helix. It is not what keeps DNA in a helix. DNA is kept in a helix by the base pairs themselves. Again, we're not going to get into it. I just want to make sure that we're not, stopping on high school definitions. It gets way more complicated than that. But again, we're not going to get into it because it's not necessary. So what is a double helix, right? So it's a two strands of nucleotides. So you have a long polymer, right? And they're exactly opposite each other, and they wind together into this helix shape, right? So it's held in place by hydrogen bonds per se. Hydrogen bonds help keep things matched up. It's very easy to break. All you need to do is heat them to about 60 to 7 degrees Celsius, and your DNA unravels. 100%. It's just going to unravel. That's how PCR works, polymerase chain reaction. You might have heard this in the news about COVID-19. Again, they use PCR tests. So they just melt. It's literally called melting. The DNA, it just means you break the double helix. It's very easy to do. That's not what gives the structure, but it does keep things in the right place, right? So we've all seen this figure before. There's our double helix. We're held together A to T, C to G. That's how things match up. And then in three-dimensional space, they kind of stack on each other. And then those stacks are actually what's holding together and what's make DNA such a stable molecule. Couple things I just wanted to mention about this, right? Most people know Watson and Crick. They get all the credit, and they got the Nobel Prize for discovering the structure of DNA. They actually piggybacked off of someone else's work. And this is why, if you ever talk to women in science type movements, Rosalind Franklin, she was the one who actually made the X-ray structure. So she shot X-rays at DNA that was crystallized, and it made a certain pattern. She was the one who initially did the experiments. Watson and Crick kind of just took her idea and ran with it. So even though Watson and Crick get all of the credit, women in science, they've been doing stuff for a long time, and they don't get the recognition that they deserve. But Rosalind Franklin definitely was absolutely critical to figuring out the double helix structure of DNA, because she was the one who actually ran the experiments to find the crystal structure of DNA. There's also been other stories. I think either Watson or Crick was on LSD when they figured out the structure for double helix. I forget which one. It's either Watson or Crick. Did like LSD, but it's never been credited to figuring out the actual structure of DNA. That was done by science, not by drugs. So this is not really a study check. You've probably done this in high school. I'm looking for complementary base pairs. What I'm asking for is this sequence that I give you is running from 3' to 5'. So maybe if I write that down. So 3' to 5'. So beginning to end. What would the complementary base DNA look like? Well, it's going to be exactly opposite. And again, it goes back to high school. A to T, G to C, T to A, C to G, G, T, T, A, C. What you might not know is because DNA is, the two strands are exactly opposite of each other. So even though the parent strain, the one that's in actual type, is going from 3' to 5', the reverse strand runs 3' to 5' in the reverse direction. And that's the way I wrote that. That allows for and we'll talk about it in a little bit, that it can be turned into proteins directly, and then also in the other direction, you can turn it into other copies of DNA immediately. Take home message, they run anti-parallel, and then A to T, G to C. Are we good on this? I know I've been saying a lot, but I do feel like we're kind of going over biology that I think most have already done before, but let me know if I'm making giant assumptions on that. All right. So let's move forward a little bit more, and then I'll give it about 5 minutes-ish, and then we'll probably turn to YouTube videos because I think it's really cool to watch. I found some really good videos that I enjoy watching. And these slides are going to lead into that. So DNA replication, right? So DNA, I think this is what confuses a lot of people, right? DNA just holds the information. Even though we say it like it's it controls the cell. Yeah, in a sense. But that's almost like saying that like the hard drive controls the computer, and it doesn't, right? It just holds the information. DNA is like the hard drive. It has all the information for a cell and actually has literally all the information for an organism, but only if we translate that. We'll talk about that in a second, but first, when a cell divides, well, how does it make a copy of itself? And that's where this anti-parallel double helix comes into play. And that's what you're seeing on the screen right now. The DNA can be split, literally, and it actually is done physically. A protein molecule comes in kind of like a fulcrum and pulls apart the DNA into two strands and then starts making two copies. And so that's why the anti-parallel part gives you exactly the same copy. One is going to be upside down, but technically, if you just flip it upside down, again, right, they would be exactly the same copy. And so that's how your body can make an easy copy. One is just the parent strain that gets basically done over twice, and then you make a new strain. So that's how you can make two from one, because both sides are telling you the same story. One goes parallel, one goes anti-parallel. But how do we get from genetic information to proteins? Proteins are the workhorse of the cell. Like, so anything that you're doing right now, breathing, moving your muscles, thinking, these are all done from protein interactions. DNA tells you or tells the body how to make the proteins, and the proteins go and do the job, right? I don't know if the book mentions it. It's an old term. It's called central dogma. Central dogma literally means DNA to RNA, RNA to protein. And so we're going to talk about that in-betweener step of how do I go from DNA to protein when I just said RNA is the in-betweener step, right? So there's multiple types of RNA, and we're going to get into that. So the three common types, there's more than this. You probably heard, maybe you heard of hairpin RNA, rRNA, what's it's up on there actually, iRNA. There's a lot of different types of RNA. The three big ones that are part of central dogma is rRNA, mRNA, and tRNA. So the first ribosomal RNA actually act like a protein. And so they make these giant complexes called ribosomes, and ribosomes are responsible for making proteins. There's a new term that was coined about 10 years ago called a ribozyme. They're technically ribozymes in that they're RNA that do catalysis. It's like an enzyme, so you call it a ribozyme. They're responsible for literally making the protein. That's all our RNA does. Messenger RNA, or mRNA, is, and I'll show you how this is done. It takes information from the DNA, turns it into a single-stranded piece of ribonucleic acid, and then that ribonucleic acid gets fed into the ribosomal RNA, and then a protein is made. tRNA is actually it's getting complicated what I'm saying. I was going to say sometimes easier to see than say. Transfer RNA is going to be a piece of RNA that's attached to an amino acid that when the mRNA goes into rRNA, tRNA comes over, they all interact, and then you start making protein. And I'll show you what that looks like. I know like my explanation can only go so far. Seeing is better than hearing. And like I said, I'll get to there in a second. I know we got about one more minute. I just want to get through. Actually, you know, we can probably cover this later because again, I think seeing is going to be better. And I'll make, you know, either we'll do this on Monday. Actually, we only have a handful of slides left. We'll get through it on Monday. So why don't I show you the videos that I wanted to show you because I think it's going to take about 10 minutes to do. And again, if you see it, I think it's going to make a little bit more sense. So why don't we do that? So actually, first thing I wanted to do is I probably should have done this at the beginning of lecture, was really briefly talk about our project that's coming up. So I had a couple of questions because people started to realize that what I was asking is easy. It is easy, but it's designed to make you think. So I gave you a list of drugs to look up and copy their structures. and I gave you the hint to go to Wikipedia. When I'm asking for the structure of remdesivir, draw me this. That's what I'm asking for. But going through slowly and drawing it yourself gives you an idea of what that molecule looks like. I also ask you to identify certain functional groups. So if I looked here, well, there's an ester. Here's an alcohol group. There's an amine, right? That's what I'm looking for in terms of the first part. It's super simple. I understand that, but I want you to get the practice of looking at actually biologically relevant molecules. Number two, before, again, I feel like I'm a little bit off topic. I should have done this at the beginning. Second part, so I want to make sure we're on the same page, is talking about going to the Protein Data Bank website. So this is the website that I sent you all in the project. So if you click on it, you'll be taken to the Protein Data Bank, or the PDB. If you just literally search in COVID-19, right, you're going to find a bunch of different proteins. That's what I want you to do. Do not use this protein. I'm going to show you just an example. I found a good one that I like. So it's CVYB. And I'll kind of walk you through what I'm looking for. So some of the questions ask you, what is the PDB code? 6VYB for this protein is the code. That's what I'm looking for. What's the name of the protein? There we go. SARS-CoV-2 spike ectodermic protein, or ectodomain structure, open state, name of the protein. What was the method? Electron microscopy. Usually what you'll see is crystal structure, X-ray crystal. That's all I'm looking for, right? I want you to think about these things. When I want different pictures taken, go to 3D view, right? All I'm asking you is for 3 screenshots. I don't care. What I really care is that you're playing with the structure to look at what's happening, right? So this is the spike protein you might have heard in the news. And so you actually can see the spike protein makes a tunnel into your cell. This is how the RNA of the virus gets into you. It literally opens up your cell. How does it do it? Well, one of these ends bind to your cells. I don't know if it's the top or the bottom, but you can definitely see it looks like a spike. It looks like a needle, and it has a hole through the center. That's where the RNA comes through. When I ask for tertiary structure, some of these have, this is quaternary structure. You can tell because there's different colors. There's 3 different colors, purple, green, and orange. All of those are different proteins. Don't worry about the boxes. Sometimes, people put weird things in their crystal structure, but this is the general structure. When I'm looking for, if I'm saying, well, how many subunits are? I look at this one, I got 1, 2, 3, 3 colors, 3 subunits. If it's a quaternary structure, that's what I'm looking at. If I'm asking for primary structure, and I said, give me the primary structure, literally copy and paste the primary structure. It's up here, right? When I'm talking about the order of the amino acids, that's the order, the primary structure. Sometimes it's hard to copy and paste directly. You might need to do a little work, but it's not that hard to find it. Copy and paste, throw it into a document, you're done. Like I said, this is designed not necessarily to give you an A or anything like that, though hopefully it will. It's designed to make you think a little. And this is all I'm asking for. I said it can be done in an hour, but I do want you to actively think.
Wait, for the primary structure, is it just one line of those letters?
That's it. I mean, you can copy and paste. It's probably going to mean multiple lines because usually primary structures for proteins can be 300 amino acids long. So it might be multiple lines, but effectively, yeah, that's it.
Okay, thank you.
Yep. Any other questions on that?
That whole thing at the top, like the lines with the primary structure.
Yep.
Is if you scroll down, is that whole thing it, or
that
is it
whole thing is the primary structure? I know. I'm just asking. I mean, that's how big proteins are. You're looking at this one protein is over 1,000 amino acids long. That's how big they are. And I do want it copy and pasted. You know what I mean? If you're sitting there typing it in by hand, you've done something wrong. Let me know if that's the problem. Just you go through the, copy and paste. If you can't find it, go to another place like Sequence. Sequence has it. You can also copy and paste from there. You can find other websites that also have the same thing. So the easiest way to do is download the FASTA file. The FASTA file will absolutely have the, I can do it right now. I just downloaded the FASTA file. There is the primary structure right there. Does that make sense?
Yes.
All right, cool. All right, so we got about 5 more minutes. I think we got about 5 more minutes of video. So what I really want to do is, well, I can skip one of them because I don't think it's that important. Two of them are really cool. It's actually going to show what's happening in real time. So why don't we watch that together? So I got two on transcription. So how does DNA become RNA? The next video will be how RNA becomes protein. So why don't we watch that? But are we ready to do that? I just want to skip ahead if we have any more questions on the project.
I'm good.
All right, cool. So why don't we watch this video?
About to see is DNA's most extraordinary secret. How a simple code is turned into flesh and blood.
And so remember when I talk about proteins constantly vibrating? This is what it's showing.
Assembling at the start of a gene.
They constantly vibrate.
A gene is simply a length of DNA instructions stretching away to the left. The assembled factors trigger the first phase of the process, reading off the information that will be needed to make the protein. Everything is ready to roll. 3, 2, 1, go.
And that's actually what happens. The other parts of DNA need to set off.
The blue molecule racing along the DNA is reading the gene.
This is happening in real
unzipping the
time
double
too.
helix and copying one of the two strands.
Not the sound, obviously.
The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to copy the A's, C's, T's, and G's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related building block known as U. You are watching this process called transcription in real time. It's happening right now in almost every cell in your body.
So that was, it's interesting to see because that's actually what's happening. It does take a complex of different proteins to come together to actually translate DNA into RNA. So what happens once the RNA has been translated? Well, it needs to be, transcribed. It needs to be translated.
When the RNA copy is complete, it snakes out into the outer part of the cell.
It looks alive, but it's not alive. It just
Then,
literally
in a dazzling
randomly
display
moves.
of choreography, all the components of a molecular machine lock together around the
This is the
RNA
ribosome.
to form a miniature factory called a ribosome.
It's made out of two pieces.
It translates
Small
the
and
genetic
large.
information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. Each transfer molecule carries a 3-letter code that is matched with the RNA in the machine.
And we'll talk about that later. It's called an anticodon.
Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecules. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second. And as a result, our muscles, brain, and all the vital organs in our body receive the oxygen they need.
All right. So I thought those were really interesting videos, and I hope you guys did too. So I'm going to call it quits on this because we've hit the end of our time. I'll put this up on YouTube later today. And then we'll finish up our very last lecture on Monday. I'm going to send out a homework assignment later today. Again, it's going to be it's similar to what we did with the Orgo homework. I've responded to a couple of people basically saying that Mastering Chem is not asking the types of questions that I want for this chapter. And so I'm going to make my own, and then I'll send those out. They're not going to be due the last day of class because it's not enough time, but I'm going to make them due by the final. So it will act as practice for these last couple of chapters. So that's my plan. All right, so if you have no questions, you guys are free to go. We'll pick up on a Monday. If you have any questions, I'll stick around for a little bit.
Thank you.
Thank you.
No problem.